Okay, welcome to the first keynote of CVPR 2024. I'm really pleased to introduce Professor Joshua Bongard, who's the Wynot Professor of Computer Science at the University of Vermont, where he directs the Morphology, Evolution, and Cognition Laboratory. His work involves automated design and manufacture of soft, evolved, crowdsourced, and biological robots. Among his many awards, he's received a Presidential Early Career Award for Scientists and Engineers, or PCASE Award, which is the highest honor given by the US government to outstanding young researchers. He was also named on the MIT Tech Review's list of the top 35 young innovators under 35, and he won a Cozzarelli Prize, which is awarded by the National Academy of Sciences to teams whose PNAS papers have made outstanding contributions to their field. He's received funding from a wide variety of agencies, NSF, NASA, DARPA, AOR, and the Sloan Foundation. He's the co-author of the book, How the Body Shapes the Way We Think. He's the instructor of a Reddit-based evolutionary robotics MOOC, and he's director of the robotics outreach program, Twitch Plays Robotics. But today, he's here with us as our keynote speaker. Let's please introduce Joshua Bongard.
Okay, thank you very much, David, for that introduction, and thanks to all of you for coming to a talk that is going to have one and only one slide in it about computer vision. I am not a computer vision expert. I consider myself a roboticist. So what I wanted to do today was to talk to you about the concept of embodiment and hopefully recruit some of you to help us design AI that designs biological or technological robots that use their bodies in particular ways to generate useful but safe behavior. David and I were just talking a few minutes ago about as an AI community, we've been so successful. We've run into a couple of issues given our success. One of them is that we're running out of convention centers in which we can host our AI conferences. The other is that some of us are thinking about where do we go next? We, and especially this community, has been so overwhelmingly successful. Where do we go with AI and robotics or embodied AI technologies in the years to come? I don't have any answers, but I have some ideas that I'd like to throw out. And again, hopefully some of you will join us in this quest to create embodied intelligent machines. So first of all, what is this concept of embodiment? So embodiment means a lot of different things to a lot of different people. But generally speaking, the idea is that having a body somehow prepares us to grapple with the fantastic complexity that the real world throws at us each and every day. This idea of embodiment has been around for a very long time. It's gone in and out of fashion over the past few decades. And if we include philosophers, then embodiment has been going in and out of fashion for centuries, if not millennia. In the current era, over the last 15 years or so, embodiment, like a lot of other ideas in AI, has been eclipsed by the deep learning revolution. But even the pioneers of AI in just the last few years have started to admit that if we want to realize true intelligence, neural networks are not going to be enough. We are going to have to house intelligence in bodies of machines so that those machines can use their bodies to push against the world and observe how the world pushes back. As we all know, non-embodied AI like ChatGPT and all the others that are out there are non-embodied, and they grapple with certain problems like common sense, cause and effect, adversarial attacks. There's lots of reasons why that might be the case. One of the reasons why non-embodied AI systems grapple with cause and effect is they do not have bodies with which they can cause effects on the world and observe how the world pushes back. In the words of Yann LeCun himself, large language models lack key capabilities necessary for achieving true intelligence, such as the ability to understand the physical world through sensory input and interaction. Roboticists have heard this request from the AI community, and they have promptly dropped deep learners into robots and hoped for the best. We've made some progress by doing so, but as we all know, organisms have a lot more going on inside than just the neural control of behavior. So how does that other stuff going on inside help organisms and humans deal with whatever the world has to throw at us? To motivate that, consider this particular example. You're looking at a developing salamander. You can see right from the beginning of development a single cell dividing into 2, into 4, and 8, and so on. I want you to try and put your shoes, not put yourself in the shoes of the salamander, but I want you to put yourself in the shoes of the individual cells as the salamander is developing. I'm going to start this slide over for you. Cells themselves are fantastically complex machines. They're fantastically complex robots. They're covered on their surface with a wider range of sensors and actuators. As you can see, they are not passive participants in development. They are active. They are on the move. They are constantly negotiating and renegotiating with their neighbors during development. This is the CVPR community, so thinking of pattern recognition for a moment, I want you to consider the patterns that each of these individual cells need to recognize and handle appropriately moment to moment during development to keep this organism alive as it transitions from a single-celled organism into one composed of billions and trillions of cells. I now want you to think about the breadth of the sensor motor experiences that these cells are dealing with during development, and I want you to now mentally sum up or compose all of those sensor motor experiences at the cellular level to the organismal level. Think about the breadth of internal experience that this animal has had before it was ever born. That internal flux prepares the organism for out-of-distribution external surprises, like the following one. You're now looking at a group of about 2,000 frog cells, and in a moment, someone is going to come in and puncture this tissue with a bobby pin. You'll notice immediately the cells react. This is, in a way, zero-shot learning. The cells immediately know what to do. They start to migrate into the wound. They change how they're talking to one another. You'll notice these brief flashes. These are calcium flashes. This is a recording of the cells talking to one another before and after this unexpected event, which they handle flawlessly. As I mentioned, these are frog cells. These frog cells have never actually been put together in this configuration before, and they've certainly never experienced this particular event before. I motivate you to puncture your laptop or your phone, and let's see how well it does. However it is that we're making smart machines, they're not smart like this, at least not yet. I mentioned that there's one and only one slide in which there's going to be some computer vision. This is it. There was some non-trivial image segmentation and registration that went into this. These particular patches of tissue, which we're going to talk about again in a moment, they like to move. This is actually a piece of tissue that's actually moving about, and it especially likes to move after it's been punctured. We have a lot of data that's reported in this paper that shows the way in which the cells communicate and react and deal with this surprise is different after the puncture event. There's much that can be learned here. Okay. So this is just a specific example of a general principle that is at work in every organism that currently exists on this planet and every organism that has ever existed on this planet, which is that every organism experiences massive internal growth, change, and surprise. And in some way, which we still don't understand very well, that morphologically pre-trains the organism to deal with surprise or change in the external world. Any of you that have done computer vision with robotics, you know that from the robot's point of view, it's constantly seeing and sensing something different and has to constantly negotiate and adapt. How do organisms go about doing that? What does that have to do with robotics? Well, obviously we've made a lot of progress in robotics over the last few years, thanks to the deep learning revolution, and thanks especially to this community. So on behalf of roboticists everywhere, thank you, computer vision researchers, for making it easier for robots to see, and in most cases, know what to do as a result. That being said, state-of-the-art robots like Google's Palm-E that you see here, are hollow inside. Not literally hollow inside, but conceptually hollow inside. This robot has never experienced any internal physical change, which every organism has. In some ways, this hobbles all robots, not just Palm-E. To illustrate this, imagine that we prompted Palm-E to look behind the drawers. This is a bit of a cooked example, but please bear with me. As you can see, Palm-E's gripper is probably too big to get behind the drawer. Its camera can't quite see. It'll probably respond to this prompt with, "I'm sorry, I can't obey this command." However, as you look at this slide, although you may not be aware of it, the motor strip part of your brain is going haywire. The motor strip part of your brain is that part of your brain that would be touched when you put on headphones or earmuffs. It's right along the medial strip here of your skull. Whether you're aware of it or not, you are actually looking at all the objects that are on the table and either consciously or subconsciously thinking about how you might obey this command. Is there anything on the table that you could reach with your own hand behind the drawer? Are there any objects here that are shiny that you might be able to move so that you can actually see or look behind the drawers? We could, of course, make Palm-E capable of using tools to respond to prompts if we trained Palm-E on a bunch of different tools. You might imagine that you are good at thinking about which objects on the table might serve as good tools because you've had 10, 20, 30, 40, 50, upwards of 50 years experience manipulating objects to get things done in the real world. That may be true, but you also have experience of being a tool and also being a tool user. What do I mean by that? Let's come back to the salamander for a moment. Again, put yourself in the shoes of the cells. As you're going to see in a few seconds, there's going to be a gap that appears in the tissue, and you're going to see that some of the cells are pushed into this gap by other cells. Some of these cells are being used as tools to see what's inside this gap, and they're being pushed into this gap by these other cells. At every level of spatial organization in the organism, from the cellular to the tissue to the organ to the whole organism level, during development, all parts of you are constantly learning about affordance detection, learning what kind of gaps are passable and impassable. How can you convince your neighbors or other things around you to go see what's inside? That pre-trains, in this case, a salamander when it confronts a gap in a rock and it can see delicious grubs on the far side. That salamander is able to use its tongue to push small twigs into the gap and pull delicious grubs out. It does it effortlessly, and it looks like zero-shot learning at the organismal level. But at the cellular level, there has been a lot of morphological pre-training. Okay, so how do we go about taking these ideas from biology and starting to build them into robotics so that those robots are not conceptually hollow? They experience constant internal change and use that to prepare themselves for external change. One way we could start to do this is to build robots from cells themselves. This is something that Mike Levin and I started doing about 5 years ago. At that time, my lab teamed up with the Levin lab at Tufts University. Levin is a biologist, and in his lab, Doug Blackiston, a staff scientist, is very adept at surgically rearranging parts of genetically unmodified tissue to create new kinds of organisms that are very far from the default organism. In this case, we're going to talk about frogs for a few minutes. Doug is very good at moving cells and bits of tissue around during development to produce an organism that grows into a healthy adult and is able to survive and thrive despite the fact that it looks very different from a default frog. Most of us are just coming off of lunch, so I am not going to show you pictures of these rearranged frogs. You'll have to use your imagination for now. 5 years ago, when we teamed up, we were jointly funded by a DARPA project. We promptly threw out the idea that we pitched to DARPA. Don't tell DARPA that. And we asked the following question. If a human surgeon can go in and rearrange bits of cells, and remember, cells are very happy to move around and be rearranged during development. If a human can rearrange tissues to produce something far from the default organism, can we recruit AI to figure out how to rearrange tissue to produce something that looks and acts very different from the default organism, but does useful work on our behalf? That led, in a moment, as you'll see, to this new field known as biobots, robots that are literally built from biological materials. You can imagine when we published this work and we admitted that we used defense funding to create AI-designed frog bots, we got a fair bit of media attention, and the public now refers to these biobots, which you're going to see in a moment, as xenobots. Xeno is short for Xenopus laevis, the particular frog that we're going to borrow materials from to make our biobots, and bot for robot. Not robots that we're familiar with like Palm-E, but these are machines that have been designed by us, or more specifically designed by an AI, to move around and do useful work on our behalf. In my book, that qualifies as a robot. Okay, 5 years ago, Doug Blackiston, the microsurgeon, told us that there were two particular cell types that he could liberate from early frog embryo: frog skin cells and frog heart muscle cells. And you're going to see Doug start to do this. So we are now looking down through the microscope with Doug. He is using microsurgical forceps to break open frog cells. So there are 6 frog cells in view at the moment. He's removing a particular part of the frog egg that contains stem cells. He's going to go after, and you're going to see him go after them here. He's going to go after those stem cells that are destined to become either skin cells or become heart cells. You can actually see the individual cells here, and throughout the next few videos, you're going to see those individual frog cells as bright white spheres. At that point, my then PhD student, Sam Kriegman, took over, and he created a biophysical model of frog skin cells, which you're going to see represented as blue cubes, and a biophysical model of frog heart cells, which you're going to see as pulsating red-green voxels. The heart cells spontaneously increase and decrease in volume, which is what heart cells do. When heart cells form up into an adult frog heart or form up into a human heart, they spontaneously synchronize and pump blood in and out of the heart. When we started tasking an AI with rearranging heart tissue or heart cells into 3D geometries that are very far from frog heart, we asked Doug, the microsurgeon, what's going to happen? Are those heart cells going to synchronize or not? And Doug's answer was, who knows, nobody's done this before. So what we built into the biophysical model was a very conservative guess. We assumed, or we told the AI, assume that these individual frog heart cells will never synchronize. So we basically are going to give the AI a very difficult design task. We then wrapped around that biophysical model a very simple evolutionary algorithm. This is a genetic algorithm. They've been around since the 1970s. What this particular genetic algorithm is going to do is search over the space of all possible ways of combining and recombining skin and heart muscle tissue. Basically, from the AI's point of view, it's a design problem where the AI can build things out of two types of Lego bricks, blue and red-green. Like every genetic algorithm, it starts with a population of random solutions to your problem. In this case, solutions are virtual xenobots. You see 3 of those initial 100 random guesses by the genetic algorithm here. We then have to give the genetic algorithm a fitness function or a loss function. We need to give it some way to measure how good these designs are at whatever we want these xenobots to do. In this first experiment, the behavioral objective that we gave to the genetic algorithm was get these bots to walk along the bottom of this virtual petri dish as quickly as possible to the right. Not a very difficult behavioral task. You'll notice that all 3 of these randomly assembled machines, not surprisingly, don't move to the right. Although if you squint very hard, and I think this is going to be tricky for those in the back, you may notice that the bottom virtual Xenobot moves to the right about a millimeter, a little bit further to the right than the other two. That's all a genetic algorithm needs, is some difference within the population. The genetic algorithm then goes to work. It deletes the poorly performing solutions, which in this case are the top and middle Xenobot. It deletes them and makes randomly modified copies of the surviving Xenobot in the lower right. By repeating this process generation after generation, we ran this on the supercomputer back at Vermont for about 2 weeks. At the end of that process, my then PhD student, Sam, got this result back from the supercomputer. According to the genetic algorithm, this was the best way that it found to put skin and heart together to make something that moves to the right. You'll notice again that the parts, or the actuators, the heart tissue, which is on the ventral or bottom half of this machine, are desynchronized or unsynchronized, but still this machine moves more or less to the right in more or less a straight line. This is just a reminder to us, and probably you, this community doesn't need to be reminded, that AI is really, really good, and in some cases, better than humans. We asked Doug, the human microsurgeon, if he could come up with a way to rearrange skin and heart muscle tissue to make something that moves to the right, and he just left the room without saying a word. So we have our AI-designed virtual biobot. Now we need to try and cross the reality gap, which is a significant challenge and an unsolved problem in robotics. How do we ensure that if we make a physical version of this thing, it's going to do what the genetic algorithm and the biophysical model predicted that it would do? So Doug went to work to try and do this. We're again looking down through the microscope with Doug. We're looking down into the bottom of the Petri dish. This big black circle that you see, this is a small concave depression in the bottom of the dish. In this case, there are some frog skin cells. They tend to be quite adhesive or sticky when taken from early embryo. You'll notice they're kind of jiggling around. They tend to stick to one another, and they're forming kind of a layer at the bottom of this depression. Doug went back and forth between looking at this MP4 that Sam sent him from the AI to going back and looking through the microscope. Doug deposited a small layer of heart cells that kind of stick together or glom together, another layer of heart, another layer of heart, layer of skin, layer of skin, layer of skin, to get the distribution of these two cell types about right. And then towards the end of this manufacturing process, Doug then takes this assemblage, reaches in with microsurgical devices again, which are nothing fancier than very, very small forceps, and is scratching away the unwanted material to make these little stubby legs and to try and approximate the 3D geometry that the genetic algorithm discovered. Just to give you a sense of scale, the Xenobot that you're looking at here, the physical Xenobot, is about 1 millimeter across. It's about the size of a poppy seed. I want you to imagine trying to look through the microscope with poppy seed-sized forceps and etching a design into that poppy seed. Doug is very accomplished at what he does. If you ever meet him, do not give him any coffee or Coca-Cola, no caffeine. Very steady hands for this task. Okay. Now the scary part came. Doug took that physical Xenobot, turned it upright, and put it on the bottom of the Petri dish. It's immersed in about room temperature fresh water, because that's where frogs are most comfortable. And at least in this attempt, we've managed to cross the sim to real gap. The physical Xenobot looks kind of like and acts kind of like what the genetic algorithm predicted it would look like and how it would behave. Again, a very, very simple robot, very, very simple task environment, but a new approach to robotics. The title of my talk is The Tip and the Iceberg. I think of embodiment as the iceberg. It can direct us in new ways and to ask new questions at the intersection of robotics and AI, and in this case, synthetic biology. So what can we do with these new kinds of robots? Well, one thing we could do is to try and recapitulate traditional robotics. As you all know, the original success story in robotics was the Roomba, the robot vacuum cleaner. Doesn't take too much to build a robot vacuum cleaner, turn towards the most dirt. In this case, you see some virtual xenobots that are doing something similar, and they're pushing some small simulated particulate matter that we put in their environment. We took those virtual bots, and Doug went to work building about a dozen of these physical bots, and again, putting them back in a different petri dish in which there's some small particulate matter, which is hard to see in this video, but you'll have to trust me, they collectively tend to push this particulate matter into piles and quote unquote clean up the dish. Hard to know what the killer apps will be for new technologies, but possibly environmental remediation in aqueous environments could be an early application of this technology. I thought for this community you might find this particular video interesting. The genetic algorithm built these robots out of skin and heart. No sensors, no brain, no neural tissue, no digestive tract, no nothing. And yet, the physical bot in the bottom half seems to be sensing and reacting to this very small bit of lint or some sort of inorganic material that's in the dish. Remember that cells themselves can see and sense their environments, they can act on what they see and sense, and they can talk to their neighbors with calcium and other messengers. So it's possible that we've created a sensing or seeing robot inadvertently, which is one of the interesting things and perhaps should give us some pause if we think about trying to create robots out of biological material. When we build technological robots, we usually get less than what we hoped for. When we build biological robots, we sometimes get more than what we were hoping for. Trying to rein in or gain rational control over sensation as well as actuation, nervous tissue, building xenobots that have brains inside. This is all stuff that you can imagine is on our to-do list. I mentioned xenobots that can see and sense. So we did actually make xenobots that can see in a way in our second paper. In our second paper, we genetically modified xenobots so that by default they would glow green, as you see here. But if they ever move about and come into contact with some phenomenon of interest, which in this case was blue light that we shone down into the dish, that blue light would genetically cause the xenobots to switch from fluorescing green to fluorescing red. And even if they then move out of the blue light, they will continue to fluoresce red for the rest of their natural life, the rest of their natural lifespan, which at the moment is about 7 to 10 days. Xenobots quote unquote live for 7 to 10 days, and then they stop moving and they rot away like any lost piece of skin tissue. Okay, so this demonstrates that it may be possible to create biological robots in future for applications in which the bots as a swarm need to go, see, remember, come back, and tell. Imagine we collected all of the xenobots out of this dish after a period of time and just simply counted the ratio of green to red bots. The swarm would in effect be reporting back how many of them came into contact with the phenomenon of interest. That was our second paper. Getting xenobots to see is an open problem. There may be some of you in this community that could help us with that. Okay. In our third paper, we asked the following question. If we can create robots that are happy to have their parts rearranged, could we realize a long-time dream in robotics, which is to make self-replicating machines? In this study that you're going to see here, we again used a biophysical model, which you see here. We wrapped that model in a genetic algorithm, and we asked the genetic algorithm to design bots that do something. In this case, that something is replicate as much as possible. Okay, here we go. So here you see one of the genetic algorithm's initial guesses. It creates a swarm of bots, in this case, 9 purple xenobots. They just happen to be spherical. And we then sprinkle into the biophysical model virtual individual frog skin cells. That's the little green cubes that you see here. And just given the sort of the way in which xenobots move, this swarm collectively pushes some of these individual frog cells into piles. As I mentioned, frog cells from early development tend to be sticky, so these virtual piles tend to stick together. We built into the fitness function for this genetic algorithm a reward if the swarm makes bigger piles. Why do we want the genetic algorithm to make bigger piles? Well, it turns out that if you make a big enough pile of frog skin cells in reality, that xenobot will start to swim. Frog skin cells, the ones that are on the outer surface of either a natural default frog or a poppy seed-sized xenobot, will grow very small hair-like appendages called cilia, and those cilia will beat against the room temperature fresh water. And if that collection of cells is small enough, all of those cilia together can in some cases cause the bot to move. So once we realized this, we moved away from heart tissue and walking robots, and we moved on to swimming xenobots. Okay, so the first task for a swarm of xenobots, if it wants to replicate, is push frog skin cells into large piles. Here's this swarm again. This particular swarm got a fitness value of 0.74 because one of the piles, I'm not sure which one of them is 74% of the way to being big enough to becoming motile. These are some measurements we made from the actual biology. A few generations later, the genetic algorithm discovered this design, or this way of making a swarm. These donut-shaped xenobots, for some reason, tend to produce bigger piles, and I'll play that for you again. These 9 donuts produce bigger piles. One of these piles is big enough to become motile. It's the one in the top left. You'll see it starts making its own pile, and that pile is big enough to in turn become motile. So in recap, what the genetic algorithm has discovered is a design for 9 bots that produce 1 child. They replicate by producing 1 child, and that 1 child replicates itself by making 1 grandchild. That grandchild doesn't produce a big enough pile to produce a great-grandchild. So this process of what's known as kinematic self-replication, replicating oneself through movement, this process of kinematic self-replication runs out of gas after 3 generations. What we asked the genetic algorithm to do is make big piles and achieve as many generations of self-replication as possible. Basically postpone the loss of replicative ability. At the end of this genetic algorithm, it found this design, which are these Pac-Man shapes. In retrospect, it's maybe not so surprising that a Pac-Man shaped Roomba, as it just moves forward, will tend to collect or make bigger piles in its forward-facing, quote-unquote, mouth. The image that you see in the upper right is showing the evolutionary trajectory. It's a visualization of the genetic algorithm finding better and more replicative swarms. Now that we have this AI-generated design, we see that the genetic algorithm is making a prediction. It predicts that 9 parental xenobots are going to make 3 children, and one of these 3 children is going to produce 1 grandchild. So again, my group at Vermont sent this particular MP4 as the specification to our biology colleagues at Tufts. And again, Doug, the microsurgeon, built a Pac-Man-shaped bot. In this case, you're only seeing 1 of the 9. And as you can see, in the way that it moves, it manages to push individual single frog skin cells into bigger and bigger piles. Those are the larger white blobs that you see here. And a few hours on from this video, this large white blob that's forming in the mouth of the Pac-Man, when we take the Pac-Man out of the dish, this blob itself will start to move and collect together enough frog cells to produce another round of replication. You might be asking yourself, why would we go about trying to make self-replicating biological robots? What could possibly go wrong? There's no good answer to that, but I will give you my attempt to answer that question. First of all, there's a lot of basic science here. We just want to understand, is this possible? Turns out it is. Whether we like it or not, the bad guys and girls out there are getting better at designing exponential technologies, and I hate to be the one to tell you, but they're using some of your technologies to do it. When it happens, and it will happen, when the bad guys and girls figure out how to make exponentially growing and spreading technologies, whether they're made of inorganic or organic materials, it's going to be up to us as the good guys and girls to design something that can defeat that self-replicating or that exponentially spreading bad technology. In my opinion, the only way we're really going to be able to do this generally and at scale is to be able to create self-replicating countermeasures in which those self-replicating countermeasures, as a side effect of their self-replication, defeat or bring down the self-replicating bad technology. Again, COVID was a bit of a warm-up for that. It took us a little while to figure out a vaccine and get it spread through the global population to nullify the threat. Unfortunately, it's likely that we're going to see repeats of that process in the years to come. Can we develop technologies like this that reduce the window between the spread of a malignant technology and the exponential spread of a benign technology that can muffle or snuff it out in some way? I don't know, but that's my answer to why would you create self-replicating frog bots funded by the US military? Okay, so that's biological robots. We've got 10 minutes left, so let's come back to more traditional robots made of non-biological components. That brings us into the realm of soft robotics. This is a burgeoning field, and I can tell you they definitely need help from computer vision and pattern recognition folks. This is a collaboration between my lab and Rebecca Kramer-Botiglio's mechanical engineering lab at Yale. Her lab is getting better and better and better at making robots out of soft parts. Not just soft bodies, but soft sensors, soft actuators, soft electronics, soft batteries, you name it. The one thing that her group, and as far as I know, no soft robotics group has managed to build yet, is a soft camera. We need your help. Again, this is a humble example, but we see a technological robot now made from technological components that's able to change internally to deal with surprise. It's not very earth-shattering surprise. We confront it with an inclined plane. Its default mode of rolling end over end doesn't work for an inclined plane, so it figures out how to change itself internally to deal with this external change. What else can we do with soft robots that are able to change themselves internally? One thing we can do, and in this case you're only going to see this in simulation, is train our soft robots to do something useful. There's a lot of agricultural and clinical applications where you might want to use soft robots. Something happens. In this case, they lose all 4 legs. There's some grievous injury, and because they can change their bodies, they recover lost structure and continue on. You've seen salamanders already in this talk. We love salamanders. They're able to grow back parts of their bodies. Maybe we can do the same with robots. Maybe we can do this with humans in the not too distant future. This is almost our blooper reel. Here you see a robot that we're going to cut in half. We assumed that this robot would try and grow back the lost half of its body, but it didn't. It surprised us and instead retracted its remaining 2 appendages and regressed from 4-legged locomotion to peristaltic or inchworm-type locomotion. Robots that can change internally sometimes surprise us in the ways that they handle external surprise. This is a very computationally intensive task. In order to simulate soft robots like the one that you see here, we have to use finite element methods wrapped inside of physics engines, wrapped inside of a trial and error genetic algorithm. It takes not weeks, but possibly months for a genetic algorithm to evolve solutions for soft robots. So in a parallel stream of research, my colleagues and I have adapted gradient descent to tune not the synaptic weights of a robot, but to tune the robot itself. Here's a screenshot. Here's some video showing you how this works. We're back to simulating a soft robot. It's basically a brick, a two-dimensional brick, if you like. We start by randomly punching some holes through the brick, which are the black dots that you see here. These black dots are going to become the raw material that the gradient descent method is going to use to design the geometry of this robot. You'll see that in a moment. The gray material that you see, this is a simulation of soft, passive silicone rubber. And the red that you see, these are small internal hollow cavities inside this brick. The red parts that are all connected, in the simulation that you're seeing in the upper right, we are pulling virtual air out of those internal cavities, which contorts the body of the robot, and then we're pushing that air back into those internal red hollow cavities, which uncontorts the robot. And by pushing and pulling air in and out of these internal hollow chambers, the robot does something. We're running this randomly designed, with this random soft robot in a differentiable physics engine. What that means is when the simulation ends every 4 seconds in this video, we compute behavioral loss. How inefficient the robot is at doing whatever we want it to do. And we can then, or the differentiable physics engine can then propagate that behavioral loss or that error back through the body and the brain of the robot and alter the synaptic weights in the robot's controller and modify these holes and these red internal cavities. What you're seeing in the bottom left is the design system obeying these gradients in air. The physics engine knows where these black dots and these hollow chambers should have been in order to reduce behavioral loss, make this robot more efficient. And this next video you're going to see confirms that fact. This is the second forward pass. And in just this second simulation, we can see that the robot is already a little bit more efficient at moving to the right. We alternate these forward simulation passes with backward propagation of error on the backward passes. And after 10 forward interspersed with 9 backward passes, we get this. You'll notice that during this process, this gradient-based method is discovering legs. It's creating a legged robot out of a piece of Swiss cheese, and it's patterning these internal cavities along the front part of the legs of the robot. This whole process, these 10 forward and 9 backward passes, takes about 30 seconds on a laptop. It took us more than 30 seconds to build the robot and push air from the top in and pull air from the top out. But again, we managed to cross the sim to real gap. I have 5 minutes left. So if you don't believe me that we can get an AI to design a soft robot in 30 seconds on a personal device, you're going to get a chance to do this right now. So feeling brave today, we're going to do a live demo with 10,000 plus people. Please get out your phones. This works better on your phone. We may end up bringing down the Wi-Fi in the convention center. We'll see. Please point the browser on your phone to the following URL. If it's too small to see, I'll try and read it off for you. bit.ly/3 uppercase R lowercase this is an eye test here. Uppercase R, lowercase k, uppercase E, uppercase Z, 3 uppercase A. Not all of you will have heard that or absorbed that, but some of you hopefully managed to type in this URL. If you did, it should take you to a Google Colab notebook that contains some code. Hands up if you got to the Colab notebook. Okay, I see quite a few hands. Okay, so far so good. I'll leave up the URL for those lagging behind. In the notebook, you should see 3 horizontal lines in the upper left. Please tap those 3 horizontal lines in the upper left. It'll give you a drop-down menu. On the drop-down menu, please tap Runtime, then tap Run All. And then the hardest thing to do, wait 60 seconds. While your robots are cooking, I'll tell you what's happening. You are now designing a robot on your phone. Actually, you are not designing the robot. The gradient descent method that I just showed you, the AI, is designing a robot for you, and it's not designing it for you on your phone. This is a Colab notebook, so thank you, Google. Google is now running this code on your behalf, allowing your AI to design a robot for you. Okay. Hopefully, some of you very soon will see a video that looks like this appearing in your notebook. This video will also be downloaded directly onto your phone. My apologies. You should find an MP4 on your phone called beforeafter.mp4. What's the before and what's the after? The upper half of this video is the before. When you started to run this code, the code generated a random body plan for you. So if you see something on your phone, look at your neighbor's phone, and they probably have a different robot in view. Everybody starts with a different random robot and a different random control policy. You can see this poor robot at the top huffing and puffing. It's running a closed-loop controller. This robot has some sensors. Those sensors are controlling the actuators. Those actuators are lengthening the springs, shown in red, and compressing the springs, shown in blue. Over this 60-second period, there were 50 forward and 49 backward passes. The gradient descent method had 50 attempts to tune the synaptic weights of the controller inside this robot, and also tune the body of the robot. It's a little hard to see, but you'll notice that the initial positions of these gray dots are slightly different in the after robot in the bottom compared to the before robot in the top. This is just meant as a souvenir for you. It's a visual representation that AI, in a non-random way, can design the body and brains of robots. If you've made it this far in this room and the overflow room, we should have a very large number of robots. If this has been successful, this was the largest crowdsourced and AI-designed robot swarm in history. If you're willing to go one more step with me, if you did obtain a before-after MP4 on your phone and you're on social media, please post it to social media along with the bit.ly link and the hashtag CVPR 2024 so we can all see what we've all collectively created in the last few minutes. I realize I'm running out of time. If you are interested in this demo, you can actually code it up from scratch. In the bit.ly link, there is a pointer to a video tutorial that'll walk you through writing this all up from scratch, and you can learn along the way the nitty-gritty details. Last thing I want to finish with is the elephant in the room, which is always the elephant in the room: adversarial attacks. We are in the process of creating autonomous technology, and unfortunately, those autonomous technologies are a monoculture. Most of the autonomous cars that are out there on the roads right now are all running more or less the same AI model, so that if somebody comes along and adds a little bit of black tape to a speed limit sign, if that manages to trick a Tesla to accelerate from 35 to 85 miles per hour, it will by definition convince all the Teslas, if they see such a sign, to do the same thing. One of the promises, I think, of these kinds of technologies that I've just shown you is we can move into an era of bespoke AI and bespoke robotics. We can task AI with creating instances of autonomous machines where every single machine is morphologically unique, they all look different, and behaviorally unique. They all do whatever it is we want them to do in unique ways. If we can create such a technological heteroculture, then by definition, they share no common Achilles heel. If an adversarial attacker finds a way to bring down one machine, that adversarial attack cannot bring down all the other machines because they all do what they do in unique ways. That's just my personal vision for one way this technology could go. Hopefully some of you can join us in that process. We are in the process of creating new kinds of biofabrication facilities that can take bespoke AI designs, unique robots, and continuously print or manufacture unique instances of those machines. Okay, I've done a lot of talking today. Of course, I didn't do any of this work. All of the work was done by the members of my lab. I'd particularly like to thank Caitlin and Thomas for the work, and Krishna for the work on the punctured xenobots, David and Sam for the original Xenobot work and the AI-driven robot design. Thanks to Rebecca and Corey and Mark at Yale for their help with the soft robots, and Doug and Mike for their biological work on the Xenobots. There's a link to my lab. I'll leave the link up if you're still trying to create an AI-designed robot. And I think it's time for questions. Thank you very much.
Thank you so much for that wonderful talk, and thanks for the souvenir as well.
You're welcome.
So we have a few minutes for questions, and there's a microphone here, and I think there's a microphone over there someplace. So if you have a question, please come to the microphone. And yeah, looks like there's a question there.
So for the robots that were collecting the frog skin cells in the Petri dish, where we've got like the Pac-Man and it's collecting all the skin cells, presumably the skin cells that it's collecting doesn't have the same arrangement of skin and muscle cells to be able to also be a robot that could then continue performing the task, right? So it's not a full self-replicating system, as it doesn't have like the manual arrangement of cells. Is that correct?
That's correct. So as you saw in the video, the children and grandchildren looked very different from the AI-designed parents. What was interesting to us is partial self-replication. It always runs out of gas. How can you task an AI with improving replicative fidelity? Make sure that the replicated machines look and act as similarly to the parental machines as possible. You're right.
So in the simulation then, when you were running it, how does the child continue to move and make a grandparent if it doesn't have muscle cells?
Sorry, in that experiment, none of them have muscle cells. They all have these cilia that beat against the water. My apologies. So they're all made of just one cell type, frog skin cells. But it turns out that there are certain shapes that can replicate different shapes, which in turn replicate different shapes.
Gotcha. Thanks for the great talk.
Sure.
And we have a question over there.
May I ask, what's your thoughts of adding like a controlling mechanism so that you can control the biological robot to, say, navigating to any arbitrary goals that you want the biological robot to go?
Yeah, the question is about control of biological systems. If you've ever raised a child or have a pet, you know how challenging this problem is. Biology has its own ideas about what it does and doesn't want to do. So it's interesting to look at these machines from a control perspective. Does top-down control even work? If we were able to include nervous tissue into these bots and have some control there, would we be able to wrest rational control over these machines? The answer is, at the moment, we don't know. It may be possible. Great question.
Thank you.
First of all, I want to thank you for one of the most mind-blowing talks I've heard ever.
Thank you.
I'm going to ask a question that maybe you can't answer because it's sort of open-ended. I want to peer into your mind, let's say 25, 50 years from now, and how you see it. You know, I'm trying to, in my vision, either it goes very wide, like you said, this notion of these super micro robots doing everything, working as a swarm, or maybe it becomes where people are 3D printing animals effectively. But you've been thinking about this a lot longer. So how do you see it 25, 50 years from now?
Yeah, I am not a biologist. I'm a computer scientist. So again, I have to wave my hands when it comes to the biology side of this. But what I've learned from my biology colleagues is it's getting easier and easier on the biofabrication facility, not to impose top-down design. So not to 3D print an animal, but to basically exploit the cellular communication that's already going on. You saw those flashes of calcium. Cells are talking to themselves all the time. They're telling each other what to do. One of the great uses of AI is to decipher what my colleague Mike Levin calls the morphogenetic code. Figuring out how AI can whisper to the cells and convince them how to build something different. So it looks likely in the years to come that it's not going to be 3D bioprinters. It's going to be AI that basically applies some external field. Could be electrical, could be mechanical, could be vibrational, that convinces cells to rearrange themselves a little bit less, a little bit more from default phenotype. Now, what that will look like in reality, who knows?
Thank you.
Let's go over here.
Okay. Yeah, hello. Sorry. Hello. Yeah. So I'm going to make this question. I think I feel it's not a really not important, but not very, it might be a dumb question, but I'm very ignorant in the theme. And at what point a biorobot is different from a living creature? You know, probably it's like, it feels a bit weird to us that because we are seeing a creature that is moving, it's even replicating. And it's kind of weird. I mean, I know it's amazing. We are just looking at skin cells, so that's fine. But at what point there's this boundary between these two?
Okay, great question. First of all, there's no such thing as dumb questions. The question is about what is the boundary between an organism and a robot or a biobot?
Yeah.
My personal view, not my professional view, is there is no boundary. This is an example of dichotomous thinking. We tend to think of living things and technology, us and them. The reality is that there is probably a smooth gradation between all of these things. Obviously, there are wild animals out there that have so far escaped human intervention. We domesticated wheat, we domesticated wolves into dogs. We've been doing this actually for a long time, tens of thousands of years, of taking the raw materials of nature and turning them to our own purposes. Until now, we've referred to them as domesticated plants and animals. But as we get better and better at stitching together different cell types from different organisms, possibly combining biological and technological components, there's a whole field called biohybrid technology, which is exactly about that. It becomes harder and harder to see where the distinction between organisms and machines lies. Actually, an interesting fact, over 100 years ago, the Czech playwright Karl Čapek wrote a play, and in that, the title of that play was Robota. That's where the word robot comes from. Basically mechanical slaves at the time, although they weren't mechanical. The organisms that were in that play, the robots, were made from living materials, not sensors and actuators and batteries. So robots are not really about what they're made from. It's were they designed by us or by AI to move about and do something useful on our behalf?
Very insightful, thank you.
Okay.
Let's go over here.
Okay, thanks for the talk. I was just feeling myself like in a sci-fi movie, right? So how do you know that the losses that you have defined in your simulation would have bring to something capable of doing not just the things that you have defined at the very beginning, but bring you to something that was doing something? Like, in other words, did you have simulation that brought you to nothing?
Did we have simulations that brought us to things that we couldn't realize in reality?
Exactly.
Yes, that happened all the time. So I mentioned biophysical models. This is a fancy word for a very simple physics engine where we tried to build in the little bit that we know about cellular motility and cell-to-cell communication. We understand 1% of 1% of what cells are doing and what they're saying to one another. So a lot of the things that the AI dreamed up was based on faulty biology or naivety about biology. So things didn't transfer to from sim to real. However, those failures are actually useful to the AI. It can indirectly bring AI to understand new kinds of biological mechanisms that we haven't seen directly. So that's a whole other branch of research going on in synthetic biology, is how do we recruit AI to learn aspects of biology that are unknown unknowns? We don't know to ask about them. We just know that the AI keeps tripping over them, and the AI can formulate hypotheses saying the only thing that explains these repeated failures is that the cells are doing this. That simple hypothesis explains all our failures. Therefore, that might be some new biology for us.
Thanks a lot.
Yep.
And I'm so sorry, we only have time for one more question, so I'll give you the last word.
Hi, thanks. I wanted to circle back on your kind of speech on the defense element. And so in the case of cellular robotics not having that Achilles heel, is that not also true for the adversarial cellular robots? And then how do you mitigate that risk?
It's a great question. The short answer is I don't know, but we are in an era in which there's an arms race, unfortunate metaphor here, that there is no proactive way to ensure nothing exponential that's bad gets out of the box. I think it's going to be, unfortunately, as we move forward, an ecosystem of good and bad things that are replicating or exponentially growing. Luckily, at the moment, the good guys and girls have more resources to throw at this and be able to design things that can bring down the bad replicating things. I don't know that there is any proactive security solution to these kinds of technologies. I hope that there are and that we discover them. Until then, we should be focusing on the best kinds of retroactive technologies we can come up with to deal with this situation. Yeah, good question. Thank you.
Thanks so much.
Okay, and so we're out of time. Let's all thank the speaker one more time. And now we have a 15-minute break before